Dear students, welcome to the LMS portal of Government of Andhra Pradesh Commissionerate of Collegiate Education. Today we will discuss about the artificial insemination and the paper 5 of zoology. And my name is G. Raviteja, working in Government Degree College, Railway Koduru, Kadapa. Here we will discuss about the learning outcomes of artificial insemination. In this introduction, the student will know about what is artificial insemination and in which kind of animals this is the process that can be used, that will be known with this topic. And the student will know the history of this artificial insemination, when it is started, and who started this artificial insemination. After that, the student will know about the which kind of persons who need these artificial inseminations. That means what kind of infertile couples are needed this kind of artificial insemination, that can be known with this topic. And what are the steps of artificial insemination, how the process will be going on, that can be known with this topic by the students. And students will also learn the types of artificial inseminations, and he will know also about the advantages and as well as the disadvantages of artificial insemination. So that's a let us we will discuss about the introduction of artificial insemination. Here, what is artificial insemination? That's the content here. It is a deliberate introduction of a sperms into the female's cervix or uterine cavity or fallopian tubes. So here the cervix is the mouth of the uterine cavity, that means womb. Sometimes the sperms are released into the cervix, and sometimes it is releasing into the uterine cavity, and sometimes it is releasing into the fallopian tube. It is the sperms are releasing in these parts to achieve the pregnancy in vivo fertilization. In vivo fertilization means the fertilization that can be takes place inside the body of woman. But some kind of other types of fertilizations are also there. In vitro fertilization, in that the fertilization can be takes place outside the body. But here in this case, in artificial insemination, the fertilization can be takes place inside the body, and it is not the sexual intercourse. That means the male and female partners, which they are not involved in copulation. Here it is. That's why it is considered as artificial insemination. We are introducing the sperms or semen into the female's body by using some kind of instruments. And here see this kind of fertility treatment, it is for not only for humans, and it is also common practice in animal breeding, such as sheep, horse, goats, cattle, pigs, dogs, and pedigree animals. And generally this is a best practice in zoo animals for captive breeding. Here in that animals, through this artificial insemination, here the animals are rising in the zoos, such as tigers, lions, turkeys, and some kind of rare animals which they are present in the zoos. And this kind of artificial insemination is also possible as tiny as honeybees, and as well as massive as orcas. Already you well known about that, killer whales. In these animals also we can see this kind of artificial insemination. And here see this, the woman who is going to take this kind of artificial insemination, after the process she get a baby. So when she get baby, for that baby she is called as biological mother. And the semen usually they are collecting from her woman's husband. If the woman's husband semen is not uh, quantitative, that means if it is not meet the requirements of artificial insemination by means of counting the number of the sperms and as well as the motility of the sperms, like that, then the semen will be collecting from the known persons like relatives, and sometimes the semen will be collecting from the unknown persons like sperm donors. In this ways, who donated the sperms or who gave the sperms to these women for the fertilization, they will be called as biological father in technical terms of this artificial insemination. but not in a natural way. So that's enough about this. And let us we'll go to the history. In this history, the first artificially inseminated animal is dog. So this was the experiment done by Italian Lazzaro Spallanzani in 1780. After 10 years, the first recorded case of artificial insemination is seen in human beings by the scientist John Hunter in 1790. In 1790, and after that, here the first reported case of artificial insemination by donor in 1884 by Dr. William H. Pankhurst. In this, as a kind of some different situation will be happen during these conditions because the professor William H. Pankhurst, he collected the sperm from the best looking from his best looking student, and the collected semen is introduced into a sedative woman. So that was the hot topic after 25 years. So before that, that case is not known by the humans. So in this way, that best looking student is unknowingly become as the first donor for donor of the semen. And let us we'll go to the next one is a sperm bank. The first sperm bank is it is started in Iowa by the in 1920 by the Iowa University researchers Jerome Sherman and Raymond Bunge. In this sperm bank, here the sperms and as well as and ova and embryos that can be preserved at minus 196 degrees Celsius. That's why this kind of process is also called as cryopreservation. Cryopreservation. So cryopreservation means the preservation of these sperms or ova or eggs at minus 196 degrees Celsius. That is called as a cryopreservation. And after that, here in UK, British obstetrician Mary Burton she started a first fertility clinic. Nowadays, so many fertility clinics are there around us, but the first fertility clinic is started in 1930s. In this clinic, the donor insemination is by the husband of this Mary Burton. He is named as Bertold Weisner, and he donated the sperm to nearly 100 members, and he is fathering up to 1,000 offsprings. In this way, he is considered as a biological father. Biological father. But here, in technical terms, it is considered as biological father, but the real fathers are different. That is about the history. And let us we'll go to the next one is who needs artificial insemination. Artificial insemination is needed for the woman with the having some type of so characters like this. So the first one is a patent fallopian tubes or open fallopian tubes. A woman normally she has two fallopian tubes that are attached to the womb. So if the among these two, if one of the fallopian tubule is a normal in structure and as well as a function, then it is better to take this kind of artificial insemination. But in some women, the fallopian tubes are damaged or blocked, or they are reduced during the development. Because of that reason, the artificial insemination is not possible. That's why at least one fallopian tube must be in working condition. And some kind of cervical factors, some women has abnormalities of cervical mucus, so that means thick cervical mucus like that. And sometimes the cervical canal is a scarring, and sometimes the cervical canal is blocked by the endometriosis. So what is that endometriosis? Endometriosis means the tissue grows outside the uterus. Tissue grows outside outside the uterus. Outside the uterus. This is called as endometriosis. If this tissue is growing outside the uterus, that may be some complicated to the uterus, fallopian tubules, and cervix. That is also altering the functioning of these parts. So, especially see this the cervical blockage is due to endometriosis. So this endometriosis sometimes it is categorized into mild, moderate, and advanced. In advanced cases, the artificial insemination is not suggested to that woman, whereas in mild and moderate conditions, this endometriosis. In that conditions, the woman is suggesting for this kind of artificial insemination. And here, sometimes the woman is having ovulatory abnormalities. So that means the menstrual cycle, which happens within 28 days. That means which takes 28 days in a normal woman. But in some women, this kind of menstrual cycles that can be takes place, it is about 40 days, 50 days, 60 days, like that, and some cases it will be in 20 days. So in such cases, the abnormalities are occurred, at which time the ovulation can be happened, that can be not known by the physician. So in these conditions, they will go for this kind of artificial insemination. And as well as in some women, they have PCOS problems. That means polycystic ovarian syndrome. In this, many cysts that can be seen in the ovaries. Due to the presence of the ovaries, the functioning of the ovary may be altered. In such kind of cases, also they need this kind of artificial insemination. And in the males, so the who is not who is unable to inseminate due to some problems, psychological problems or mental problems, or sometimes morphological problems in the testicles or in the tubules of the testis. So it is unable to inseminate. In such kind of males, this kind of method is suggestible. And in some kind of women, they inseminate, but the sperms in that semen is completely absent. That kind of condition is called as azoospermia. In such kind of cases, the semen will be collecting from the donor. So donor semen will be taken in this process. In this way, they will have the babies. And in some kind of cases, same sex couples, they also need this kind of artificial insemination, but it is seen in the same kind of sex in females, but not in males. So sometimes homosexuals, the uh, male can with a female with a female only, like that, they are living together in such a kind of relationship. In such cases, if that women's they need this kind of if they need a pregnancy, if they need child, they will go for this kind of artificial insemination. That's it. Let us We'll go to the steps in artificial insemination. There are 5 steps are there in this artificial insemination. Let us We will discuss about the first step, that is the physical examination. In the physical examination, both the couples are completely examined. So the female, first of all, she examined with the help of hysterosalpingography. In this hysteroscope is introduced into the body to check the uterus, and fallopian tubes, is they are normal or not, and cervix. This can be checked with the help of some kind of images taken by the hysteroscope. So this hysteroscope is inserted into the female's body, and they check the functioning and the structure of this uterus, and as well as the fallopian tubes and cervix. And whereas in this female, some ultrasounds are also used, ultrasound scanning. In this ultrasound scanning, they check the ovaries, and inside the ovaries, they will check the status of the follicles, is they are growing or not in a normal way. That can be checked, that can be screened in a woman completely. Whereas in the males also, in the case of males also, this ultrasound scanning is very useful to know about the functioning of the testis, and as well as the tubules which are associated to the testis, to the penis. So in this way, the first screening can be seen. And after that, blood tests. During these blood tests, they can assess the hormone levels in the body. So if the hormones are releasing in a proper manner or not, that can be assessed through the blood testings. And the next one is a sperm analysis. In this sperm analysis, normally a person ejaculates 3.5 ml of semen in an ejaculation. So that is the normal ejaculation in a normal man. So in this semen, 3.5 ml of semen. If this semen is with 15 millions of sperms per 1 ml, 15 millions of sperms of 1 ml, then he will that means that semen is usually used in this artificial insemination. If the sperms present in this semen are with they will also check for morphology. That means the sperms which they are having a normal morphology that can be checked in this sperm analysis. And motility of the sperms is also assessed. If the motile sperms are not there, it is not possible for fertilization in this artificial insemination. That's why the sperm analysis is placing a major role in this artificial insemination. If a person is with a oligospermia, azoospermia means complete absence. Oligospermia, zoospermia. In that kind of cases also, this artificial insemination is best for that to have the babies. So by having repeated collection of the semen, they collect a more number of motile sperms through these steps. And after that, the second step is fertility drugs and monitoring. During these, some kind of drugs like clomid and gonadotropins, which are injections which are given to the female body before the AA procedure. These kind of hormones that they stimulate a pituitary gland. Already you well known about this pituitary gland. It is the master gland. Then this pituitary gland is activated and release two kinds of hormones which are essential, that is follicle stimulating hormone and a luteinizing hormone. The follicle stimulating hormone is helpful for the growth of the follicles, whereas the luteinizing hormone is helpful for the ovulation. That means releasing of the ova from the ovary is helped by these hormones. That's why to monitoring these levels of these hormones, they should be given this kind of clomid and as well as gonadotropins. And next, human chorionic gonadotropin is given to the woman to trigger the ovulation and for the maturation of the follicles also. In the insertion of the sperm within 24 to 6, 34, 24 to 36 hours after this injection. That means here the logical thing is, after this injection, then within 24 to 34 hours, the egg will be releasing from the ovary. That's why the sperms are introduced into this female body. So that is the logic here it is. And next, closely monitored with blood tests and ultrasounds. Blood tests which they are helpful to check the hormone levels, hormonal, hormonal, hormone levels. And ultrasounds that is basically helpful to check the follicle size in the ovary. So it is growing or not. If it is growing, is it ready to release or not? That can be assessed through these ultrasounds. And after that, so tracking of ovulatory cycles, this is monitoring by the physicians. So they are monitoring the ovulatory cycles regularly. For what basis they are monitoring? Here it is this cycle is a normal cycle or induced cycle, and at which time the ova is releasing into the body, and is the endometrium is developing or not, that can be checked through this tracking of these ovulatory cycles. And after that, testing for LH surge in her urine. LH means luteinizing hormone. This luteinizing hormone is responsible for ovulation. Ovulation means releasing of ova, releasing of ova from ovaries. So that ovulation can be takes place when the LH levels are high in the blood. That is called as LH surge. So during if these LH levels are more, when we are checking for the blood tests, then immediately they are going to insert the sperms. So that is the conditions. And during this surge times, one or two artificial inseminations are given. not only a single artificial insemination. In some cases, more than one artificial insemination procedure can be done in this time. So that is about the fertility drugs and monitoring. Let us, we will go to the next step, that is preparing the sperms. In this, the sperm sample is collecting from the desired persons, or from her husband, or from the donors, unknown persons or known persons, like that. After the sperms are collected from the person, that should be washed and processed. Why the washing and the processing phase can be seen here in this washing and processing, the toxic substances are removed from the body. Toxic substances removed from the body, and as well as a shell around the sperm. Around the sperm, some gelatinous cover is present, that is also removed through this process. When we remove this kind of contents, then it is easy to uh, it is helpful for the success rate of the fertilization. And let us see this. If you will introduce this unwashed semen, then it may elicit some kind of problems like uterine cramping, expelling the semen, and causing pain due to prostaglandins. Because why? What is the reason? So these prostaglandins are some kind of toxic substances, but actually they stimulate the myometrium of the uterus. Myometrium, the muscles in the endometrium. So these muscles somewhat irritated by these prostaglandins. At that time, then uterine cramping is occur due to the vigorous contractions of the muscles, and sometimes it may be helpful in expelling the semen. What we introduced, the semen introduced into the female's body in this process, artificial insemination, that can be expelled due to the presence of this kind of prostaglandins. And some pain also occur due to the contractions and relaxations of these muscles. That's why we should you introduce the washed semen into the womb of the woman in this artificial insemination. That is better to avoid the problems. And in the case of husband's sperms are not meeting the requirements of this artificial insemination, then we are going for donor sperm. That means in counting the number, and as well as motility of the sperms, and morphology of the sperms, if it is normal or usual, then they will go for donor sperm. But after the collection of the donor sperm, it should be quarantined at least 6 months. Why it is happening? After the collection of the sperm, then it is checked, it is screened for infectious diseases like HIV, herpes, and hepatitis, like that. So in this, after 6 months, in during the incubation period, some kind of diseases may be appeared. Because of this reason, the quarantine is necessary. After the quarantine, again the semen will be screened for infectious diseases. That's why this quarantine time is essential to avoid the screening infectious diseases from donor to the that woman. And the process of sperm washing offer several benefits. Let us we will look into that several benefits. What are that? The sperms are concentrated to boost the chances of getting pregnant. So here the sperms in this the shell is removed. When the shell is removed, then they are actively moving in the medium, that means a nutrient medium. That semen, when it is introduced into the woman's womb, then they are quickly moving towards the egg, which is the present in the fallopian tubes. In this way, it boosts the chances of getting pregnant. And after that, it may prevent the female partner from the allergic responses. Due to the presence of toxic substances, some kind of allergic responses can be happen in the female, that can be removed, that can be prevented in the woman due to the processing and washing of the semen. And it may help to minimize any uterine cramping because of the removing of prostaglandins. That's enough about this. Let us, we will go to the next step, that is insertion of sperms. It is very easy process and quick process, and it is a painless. So and this, when the procedure begins, the female partner will lie down on a bed for pelvic examinations. After the pelvic examination, then a vaginal speculum is inserted into the body. This vaginal speculum is helpful for widening of the vagina. It is helpful for widening of vagina. So here in this case, later the sperms are placed in all the way into the uterine cavity with a thin catheter and a syringe. The syringe is gently pressing to release the sperms. That's it. And let us will go to the last step, the most important step, that is rest. The rest is needed after the artificial insemination. So that is about half an hour. So after the artificial insemination, a cervical cap or sponge is placed. See this is the cervical cap. This is the cervical cap or sponge which is introducing in the artificial insemination. Sorry, introduced into the cervix region. So that is to avoid the leakage of the semen from the cervix to the outside of the body. And the woman may be advised to lie down for 15 to 20 minutes. In some cases it may be to half an hour. It is to boost up the success rate of the fertilization through this artificial insemination. And after that, it is to help for the more relaxing of her. She may be bring her, she may want to bring her partner or close friend, or a book, or a game in a mobile, whatever it helps to relax, that can be taken in this process. Later here, if she is still unable to get the pregnant after several cycles, the doctor may want to do further fertility testing. So, and is it suitable for that woman or not, that can be also assessed here in this. If it is not, means she advised to take for another kind of processes. And let us we will see this process through an animation series. In this, this is the female reproductive system, and in this, this is the part that is called as a vagina, and after that this is called as a cervix. This is the mouth of the uterus, and after that the pear shaped structure, this is called as uterus. This is all, this is called as uterus. And this is called as ovary. And here inside the ovary, these are the follicles which they are developing. And this is the fallopian tube. You well known about this. In this fallopian tubule, we must know about a part that is ampulla, the swollen region of the fallopian tube, that is ampulla. In the ampulla, the fertilization can be takes place. Let us we will look at this process. This is the follicle. It is gradually maturing, and so it is gradually maturing like this. After the maturation, it is ruptured due to the LH surge, and when the ruptured ova is released into the cavity, peritoneal cavity, later it is collected by the fimbriae of the fallopian tubes, and it is transformed to the ampulla of the fallopian tube. Later, a vaginal speculum. This is the vaginal speculum. That is the instrument. This is helpful for the widening of the vagina, that is inserted into the vagina. It is to widening the vagina. And after that, a catheter is taken. Here it is the catheter, actually the needle like structure, that is a tube, a thin tube, uh, uh, transparent one is called as a catheter. Unless next, this catheter is connected to a syringe. With the help of the syringe, they collect the semen, that means which are healthy sperms are collected with the help of this syringe that is connected to the catheter. And this catheter and syringe is introduced into the uterus through the speculum, and later slowly the syringe is gently pressing to release the sperms which they are migrating from the uterus to the uh, egg by swimming, and as well as the contractions of the uterus. And some kind of sperms may stop, and some kind of sperms may enter the ova and fertilize them in the ampullary region. And after that, after the completion of this process, lastly the cervical cap, this is called a cervical cap or a sponge, that is placed in the cervical region. So this is for 20 to 30 minutes. After that, it will be removed. The cervical cap is to prevent the leakage of sperms which are released into this area. The sperms which are released in this area can be leaked from this area. To avoid this leakage, the cervical cap is inserted in this region. So that is about the process of this artificial insemination. And we will look into these types of artificial insemination. In this case, the first one is intracervical insemination. That means the semen is introduced near the cervix or into the cervical region. That's why it is called as intracervical insemination. And the second one is intrauterine insemination. In this, the semen is releasing into the uterus, not in the cervical region. So, but this intracervical insemination that can be takes place at a home itself by women. But it is unlike that. It is not doing like that. It need a practitioner. Healthy practitioner can be needed for this kind of intrauterine insemination, and it is the most successful process of the four types of this artificial insemination. And let us we'll go to the third one is intrauterine tuboperitoneal insemination. That means the semen is created, 10 ml volume of a semen is created, that is introduced into the uterine and as well as in the tubes, fallopian tubes, and finally introduced the semen into the peritoneal cavity present around the ovary. That's why it is called as intrauterine tuboperitoneal insemination. And after that, if the sperms are released into only the fallopian tubes, that is called as the intratubal insemination. Among these four types of inseminations, the best one is the intrauterine insemination. It is widely used in many fertility clinics. And let us we'll go to the then we will discuss about the advantages of artificial insemination. So many advantages are there during this artificial insemination because there is no direct contact of male with the female partner. So if male is having some kind of infectious diseases that may be transformed to the female during the contagious or due to the contact of this sexual contact, that can be avoided in this artificial insemination. In this, the first one is a disease control. So this disease control is happening due to the screening of the sperms, screening of semen. So that screening is for infectious diseases like HIV, hepatitis, and the sexually transmitted diseases or venereal diseases that can be avoided through this artificial insemination. And next one is semen can be collected from the desired persons. For example, a woman in the social media asked the sperms or the semen of the Hrithik Roshan, but he rejected that. But actually, so in this manner, in this technique, it is a possible way to collect the to take the semen from the desired persons if they are willing. So that is the one of the advantages. So some of the women they need the semen of the skilled persons, and some of the women they need the semen of the handsome persons like that. So that is, and next the semen is evaluated each time to be which is collected. Why? Because the it is evaluated for the screening, screening of infectious diseases, and as well as motility of the sperms and counting the number of the sperms. So otherwise the artificial insemination may be failed. That's it. And next, relief from the sperm allergy. So in this the semen is completely washed and processed, and in this process the toxic substances are removed from this semen. Then they will relieve there is a relief from the sperm allergy to the woman. And after that, a solution for cervical issues. The artificial insemination is a solution for cervical issues as I discussed previous. And important persons also they will go for this kind of artificial insemination. Male infertility and low sperm count issues and unable to ejaculate the semen. Those are the in that kind of cases. Then they will choose for this kind of technique, that is artificial insemination. Let us we will go to the next one is the disadvantages of artificial insemination. In this, Here this is an expensive process, and a well equipped laboratory is needed to do the various kinds of screenings, and as well as to this process also. And an expertise, a technical expertise is also needed for this process, except for intracervical insemination. So for the remaining processes are this kind of technical person is needed. So that's why somewhat it is disadvantage. And next, incorrectly placed artificial insemination. That means if the practitioner or expertise is not doing well, then that causes serious problems like a hormonal imbalance and internal bleeding to the woman. So that may to cause some kind of syndromes also. So thank you to one and all.